Hello everyone, welcome back to my YouTube channel. This is Chaitra, studying in Sri Venkateswara College of Engineering, Bangalore. I am currently pursuing my BE in Computer Science and Engineering Cyber Security. Today's subject is Biology for Engineers, and I'll be talking about human blood substitute. Let's get started with the video. As mentioned earlier, today's topic will be about human blood substitute. Let's get started. So, let's have an introduction to human blood substitute. Human blood substitutes are designed to mimic the functions of natural blood that developed to address the limitations of the blood transfusions, including shortages and disease transmission. Understanding these substitutes is crucial for advancing medical treatments and emergency care. Now, let us understand the importance of blood substitute. Blood substitutes can potentially save lives in the situation where blood supply is limited. They may also reduce the risk of transfusion related infection and immune reaction. The development of these alternatives is essential for military and disaster response scenario. The types of blood substitute, it can serve as two types: plasma volume expanders and replicate of oxygen carrying function of the natural blood. Plasma expanders, these are the compounds which are either entirely synthetic or processed from the natural proteins that serves as infusion solution which expand the intravascular volume. RBC substitute, modified hemoglobins. These are essentially human hemoglobins extracted from the outdated blood. Perfluorocarbons, synthetic organic compound that can take over perfusions. Types of blood substitute. There are primarily two categories: oxygen carriers and plasma expanders. Oxygen carriers like hemoglobin based and perfluorocarbon based substitute are designed to transport oxygen. Plasma expanders, they aim to increase blood volume and maintain circulation during the emergencies. Now let us talk about hemoglobin based oxygen carrier, HBOC. Hemoglobin based oxygen carriers are derived from the human or animal hemoglobin and are modified for safety and efficiency. They have been shown to efficiently transport oxygen to tissues, especially in anemia cases. However, concerns remain about their side effects, including vasoconstriction and organ damage. Perfluorocarbon based oxygen carrier, PFC. PFC are synthetic compounds that can dissolve and transport oxygen and carbon dioxide. They are particularly useful in environment with low oxygen level such as high altitudes. Clinical uses of PFC is still under investigation with challenges related to delivery and metabolism. So, as studied in previous slides, we came to know that there are two types of blood substitute. In that, the first type is plasma expanders. Now let's start with plasma expanders. Plasma expanders such as dextran and hydroxyethyl starch help to maintain blood volume during the shock. These substances are designed to enhance fluid retention within the vascular system. While effective, they may lead to complications like allergic reaction and kidney damage. Current research and development. Ongoing research focuses on improving the safety and effectiveness of the blood substitute. Advances in technology have led to the new formulation that aim to reduce the side effects. Clinical trials are crucial for determining the optimal use of the substitutes in various medical conditions. Now let us discuss the regulatory consideration. The approval process for blood substitute involves rigorous testing for safety and efficiency. Regulatory agencies like FDA and EMSH stringent the guidelines for their development. Ethical concerns regarding their use, especially in emergency setting, must also be addressed. The consideration also include guideline on the similar biological medicinal product, guideline on similar biological medicinal product containing biotechnology derived protein as an active substance, quality issues. Guideline on similar biological medical product containing biotechnology derived proteins as active substances, quality issues, non-clinical and clinical issues. Recombinant erythroproteins, low molecular weight, have parents, recombinant interferon alpha, recombinant GCFS, somatropin, recombinant human insulin. Now let's start with advantages of blood substitute. Blood substitute can reduce reliance on human blood donation, addressing supply shortages. They have a low risk of transmitting infections compared to traditional blood transfusions. Ability to be manufactured in a large quantities make them a reliable option for emergency medical situations. Now let us talk about the limitation or the challenges faced during the blood substitutes. Despite their benefit, blood substitutes can sometimes lead to adverse effects such as increased blood pressure. The long term effects of using blood substitutes in patients are still not fully understood. Regulatory approval processes for new blood substitute can be lengthy and complex. Thank you for watching my video. Please do like and share for more interesting videos. Thank you. Now let us understand the future aspects of human blood substitute. The future of human blood substitute hold promise for enhancing patient care during the critical situation. Continued innovation may lead to safer, more effective alternatives that can be widely used. Collaboration between researchers, clinicals, and regulatory bodies will be essential for the success in the field.